And also, I would like to thank the Vebleo and entire committee for giving me this opportunity to speak about what we are doing here at UT Tyler. Basically, I recently moved to UT Tyler, but most of the research that I'm going to present here, which we are continuing from Kansas State University. And today's topic is engineering extracellular vesicles as a magnetic resonance contrast agent, actually. So here in this topic, I will be trying to share some of the exciting work that we have been continuously working on, the utilizing the cellular biologics in the drug delivery in the broader spectrum of nanomedicine. Before going towards my topic, I would like to briefly share, well, she has already been shared lots of the things. I would like to briefly share my career journey. I started my career, as an organic chemist back, 1990 to 1997, and I was teaching in T1 University in Nepal until 2004. So I did my master's in organic chemistry in T1 University. Since then, I was teaching in the same university as a chemistry lecturer. Later, I got an opportunity at Santosh University for my graduate studies, where we started to work on the, utilizing the bio nano system engineering to address the various, challenges that we are facing day to day in the health science. Uh, Completing my PhD at Santosh University, I started my first postdoc at University of Wisconsin, Milwaukee, where I was working in the polymer synthesis and nanoparticles. Thereafter, I moved to UC San Diego, at Morse Cancer Center. That is the place where I started to work with the clinicians and understand the clinical language per se. So how I can bridge as a brain scientist, but there is a language gap to understand that when we go for the application. So I was trying to develop myself as a bridge scientist who can make a communication between the brain scientist as well as the brain scientist. So nearly 4 years, I enjoyed working at Morse Cancer Center. Then after I moved to Houston Methodist Hospital, where I was working in the translation imaging department in Nanomedicine group. So this is the place where I had an opportunity to explore the nanotechnology in the imaging. Then I started Kansas State University 2014 to 2021. And recently I moved here. I have been running the small group of 6 people, great graduate, undergraduate students and postdocs. I had a very great time, and the same research program right now here at UT Tyler. So speaking of the research program that we are working in our group is a broader spectrum of organic chemistry, material chemistry, and nanomedicine. So we do the synthesis of the small molecules or phospholipid polymer chemistry, and we use the chemical products synthesized in our laboratory to engineer the nanomaterials for biomedical applications. For instance, here you can see that multifunctional nanoparticles, as well as how we can use the cellular properties to tune the nanomaterials for the biological applications. Similarly, since we are using lots of polymer chemistry and polymer synthesis, we also do some work on the tissue engineering. And this is a pretty typical example where we design the broad vasculatures using polymer. And once we have this engineered construct, that we move forward to understand their biological application possibilities in drug delivery, imaging, and tissue engineering. So within this background, our laboratory is entirely working in the broadest spectrum of nanotechnology. So since it is a nanotechnology, I would like to briefly go into what is nanotechnology, how US nanotechnology initiative defines the nanotechnology. The research and technology development at the atomic, molecular level in the length scale of 1 to 100 nanometer. So when we say 1 to 100 nanometer, is to give some idea about the precision of this scaling. The water molecule is about 0.1 nanometers. You can see the size of tennis balls. So when we talk about nanotechnology and nanomedicine or drug delivery system, we are talking about 10 to nearly 200, 250 nanometer scale range. So the materials I will be presenting here with you will be around the size of 100 to 200 nanometer size. So this nanomedicines is the branch of nanotechnology, which is for treatment, diagnosis, monitoring, and control of biological system. So overall, here the idea is how we can do all this application in biomedical science using a single technology. So the aim of nanomedicine to devise all these extraordinary features in a single packet so that we can minimize the cost, we can minimize the treatment time, we can minimize the toxicity and many more. While speaking about the medicine itself is not new. We have a great history of medicines. You can see here, back in the recorded history, we have back in 3000 BC, we started to treat our patient using religious empirical. Maybe in the community, some people, which is the senior of the community, define the religions, and then we follow the religious guidelines to treat the patient. Then we started to improve our, basically our understanding. We started to use natural empirical and the religious authority, observation empirical. We started to observe what is the cause of disease actually in the 1500 AD. So we got our first, antibiotic in 1920, where this is the era, 1850 to 1940, where we started the molecular medicine. Okay. So it has been expected that, by 2010, the nanomedicine will be in the market. But we all peoples, scientists around the world working so hard that our first nanomedicine was approved by US FDA, which we call Doxil, in 1995. Now we have plethora of the nanomedicine. You can see here, we started to learn about liposomes in 1964. And we got a Doxil, which is the liposome, a lipid bilayer encapsulated doxorubicin, which was approved in 1995 by US FDA, first nanomedicine. Then after we got Abraxane, which is the protein-bound paclitaxel, another chemotherapeutic agent for cancer. And now we have many more drugs, nanotechnology drugs, in the market, which were approved in United States, Europe, and around the world, and nearly hundreds of. I think we need to update this list. I need to update this list further. So now, great example of nanotechnology, nanomedicine, the recently, coronavirus, the Pfizer biotech, Moderna, which is the lipid particle encapsulated mRNA. So this is the outbreaking, and this can show that the importance of the nanotechnology in coming eras. Okay? So what is the advantage of this nanotechnology? Why we are talking about nanotechnology? This is also very important to briefly understand what the nanotechnology, where the nanotechnology is taking us, what are the advantages of this nanotechnology. So the first advantage of nanotechnology, the multifunctionality. Okay. The second example is the unique electromagnetic property. When the materials in the nanoscales, when we cross down the materials from ball to nanoscale, we change their magnetic properties. Similarly, this nanomaterials can be, or nanomedicine, or this drug delivery system can be tuned in such a way we can take an advantage of the pathology of the disease sites. We'll discuss this functionality each one by one. I think I have more time today since the second is first speaker is unfortunately because of time schedule. So in multifunctionality, we are talking about the nanoparticle here. So these nanoparticles, when the size is nano, the surface to volume ratio will be increased. The first functionality while increasing the surface to volume ratio is the solubilization. There are lots of drugs, small molecule drugs, which are not soluble in water. And we are not happy to inject these organic solvents to make this drug solubilize in our body. We want a really body-friendly, aqueous environment so that our body will not, have any adverse effect. The solubilization. The great example for this solubilization is the hydrophobic paclitaxel drug, which is solubilized in the protein nanoparticle Abraxane. So this Abraxane is US FDA, the great example of solubilizing drug. The second multifunctionality is the protection from the degradation. So the great example for this one, the patisiran. This is siRNA encapsulated in the nanoparticle, for therapeutic purpose of siRNA. These RNAs are very fragile. They basically degrade within the body once we inject. So this nanotechnology helps to protect these, RNAs to getting degraded. The third multifunctionality, that is the immune evasion. So whatever we inject for the treatment purpose, our body, we have our own defense mechanisms, which I will be taking this defense mechanism example to design these nanomaterials in later slide. So whatever we inject, our body will recognize the foreign material and it immediately wants us to excrete out, right? So therefore we need to hide this small molecule drugs, even the nanotechnology itself, nanomaterial itself, from to make it body friendly. The great example is the doxil, which we talked about. Doxil is the it's a lipid vesicles which is coated with polyethylene glycol, water soluble polymer. So this water-soluble polymer creates the hydration layer on the nanoparticle surface. So this hydration layers cooperate with the immune system so that this drug, this nanomedicine, can circulate prolonged period of time in our body. Once it circulates prolonged period, once we can able to tune the pharmacokinetics of this nanoparticle or drug itself, then we'll have opportunity this drug can go to this target site. That's a great example. Similarly, the targeting. So right now we don't have any targeted drug delivery system approved for US FDA, but there have been great ongoing effort. For instance, BIN01, which is already in the second stage of clinical trial. So this is also docetaxel nanoparticle, which target to the PSMA, the prostate specific membrane antigen, which is at the third stage of the clinical trial. Right now, we don't have any nanomedicine or drug delivery system that is approved, which is targeted. Similarly, imaging agents, we can tag the lots of imaging agents in these nano materials, which we will be discussing in later slides. Drug combination. In addition to the single molecule drug, we can use the multi drugs so that we can overcome the drug resistance that we have already seen in cancer. Not only cancer, also in the bacterial resistance can be overcome. One great example is the CPX, liposomal catarabine and daunorubicin, which is used in leukemia. This is the 5:1 combination ratio of these two drugs. This is also the lipid vesicles. Similarly, the trigger activation. So we can also take advantage of the physiology of the pathological side, the disease side, and use that physiological environment, the pathophysiological environment to trigger the drug release. So we don't have any approved. I haven't seen. Maybe I might need to go more for deeper. But there is the CD166 Pro, and the antibody targeted drug CX2009, which is in the phase 2 clinical trial. So what happens once this nano drug get into that site? So the CD166 probody that started to trigger the cut this pro drug, and active drug will be released. So we can take the advantage of this pathophysiological. That's the multifunctionality that we can package in a single nanotechnology, single nanomedicines. That's the advantage of nanoparticle. So similarly, second advantage is unique electromagnetic properties. When the materials in the bulk stage and the same materials in the nanotechnology, nanoscale, their magnetic, optical, and thermal electronic properties will change. The great example for this one is the nanotherm therapy. This is the iron oxide nanoparticles used to treat this tumor in the brain, and also we can track using the magnetic resonance imaging the therapeutic potential. Similarly, the third advantage of nanomedicine, which we already discussed, is the taking advantage of pathological site. For instance, tumor. Let's take an example of tumor. The tumor tissue is not only a cancer cell. It's lots of different types: immune cells, fibroblasts, stroma cells, hypoxic region, different cells as well as different compartment in this tissue. For instance, if you go from away from the blood vessels in the tumor tissue, you'll see the increasing interstitial pressure. We can take the advantage of pressure. Similarly, tumor environment is highly acidic. We can take that acidic environment advantage to drug delivery. Similarly, there is a hypoxia. We can also take the hypoxia environment to tune the kinetics of the drug. So using this nanotechnology, these are the major applications that what nanotechnology. So we have been grown a lot, and also I have shown you that there are numerous nanotechnology drugs were already in the market, and of course, we really need to acknowledge all the chemists as well as pharmacists and all the scientists working in the small molecule therapy, small molecule synthesis. There have been lots of small molecules that we are using in our medical applications. So some are water soluble, some are water insoluble. So they need some, maybe there are the opportunity that we might need to revisit the formulation of these drugs so that we can make these drugs our body friendly. So while we have been improving a lot in cancer per se, but if we see the statics of the cancer, this is so deadly. You can see here, so I do have statics until up to 2015. If we see here, the cancer is nearly plateau. We have some incline in the cancer. We are, because the technology had advanced, so we are starting to, we have a great success in early, breast cancer, early ovarian cancer, early detections and all so on. But if you see in the heart disease, we have declining a lot, because in heart disease, we can change our daily habit, which we can change our basically, physical habits, and we can control the disease. But cancer is abnormal growth. If you see this statics here, this is further deadly, and it scares us. The expected, cancer cases as increasing every year. So what is the major cause of this? Our body is the major cause, because the limitations in the cancer therapy, cancer is heterogeneous mixture of cells, and which is secondary growth. The problem is our body. We have our own defense system, that is immune system, that also protect the drug to be getting into this tumor. So tumor is some way on the deep organ here. We are injecting drug from here. So this drug need to go with all these physiological barriers, as well as all these organ barriers, phagocytic organ barriers or excretion barriers. So there are different, these barriers, our drug, small molecule, not only small molecule, even the nanomedicine encounter this obstacle. So once, if we reach into the tumor, so tumor is heterogeneous, right? We can see lots of cells are here, lots of different types of cells. And how we can recognize the cancer cell in the heterogeneous mass of the tumor? That's the biggest question. For instance, if we recognize the cancer in this heterogeneous mass, then again our cell, we have cellular immune system. Cell also has a defense mechanism. So whatever internalized into the cells were wrapped by endosome or lysosome membrane. These are highly acidic and they are highly rich in different types of enzymes. So they basically degrade this medicine. So how we can selectively protect this medicine from this intracellular degradation? So that's the case. That's the reason that what the nanotechnology we are talking about, and how we can cooperate with this intracellular environment as well as intra-tumor environment to trigger the drug precisely into the cancer cell. So again, we need to think about this heterogeneous tumor mass here. So in this heterogeneous tumor mass, our circulating immune cells does attack this very efficiently, right? While they also compromise themselves and becomes the tumor-associated immune cell. For instance, tumor-associated macrophage, they further enhance the growth of the tumor. So there are lots of limitations there. But now we have our immune system that naturally seeks for the cancer cell or any inflammatory cell. For instance, these are the immune cells that are the soldiers of our body that basically fight against the disease or any inflammation in our system. So one of these is our interest in the natural killer cell that naturally seeks for the cancer cell, or basically they try to find the all inflammatory or different foreign materials and try to kill that by helping this taking advantage of different types of receptors present in this natural killer cell, which naturally seeks for the cancer cell. One example I would like to show you in this video here. So these bright dots are the natural killer cell, NK cell, and these are the cancer cells. So when we incubated this, when we treated this cancer cell with this, natural killer NK cells, they destroy the cancer. They naturally seek for the cancer and destroy the cancer. So taking this knowledge, we started to taking advantage of immune system and try to re-engineer the nanomedicine in such a way that it looks like our immune system. so that they can naturally go and find the disease or cancer cells or disease or inflammatory cells. Okay? For that, we can re-engineer. We have a great example of CAR T therapy, right? So which is re-engineered T cells, so we that injected back to the patient. Excellent example. But the question is, the population of this immune cell, for instance, NK cell or tumor cell in our circulating blood is very, very small. And extraction and re-engineering process also highly tedious. But the good thing is these cells also releases the vesicles called extracellular vesicles. Numbers of thousands of numbers of extracellular vesicles released from the one cells. But these extracellular vesicles carry the parent-specific properties. That means that these extracellular vesicles also have an ability to communicate with the disease cell. Indeed, that these extracellular vesicles are already people are already using to target different types of disease. So one example, so taking advantage of this extracellular vesicle instead of the whole cell, we are starting our engineering technology to devise these extracellular vesicles with the synthetic nanoparticle, because the yield of extracellular vesicle is very low. But if we can hybridize this with the synthetic materials, we can enhance the number of synthetic materials. They are behaving like extracellular vesicles, and extracellular vesicles have the parent specific signatures that basically seek for. For instance, the natural killer cell derived extracellular vesicle should naturally seek the cancer cell as shown in this video. So speaking of extracellular vesicles, these are exactly looks like liposomes. The differences here is this vesicles has a bilayer, and this bilayer has a different types of signature proteins. Because of these signature proteins present in this extracellular, they are unique. And these signature proteins properties are also resembles with their parent cells, and also has a lots of different biological molecules that trigger the communication, as well as that trigger the, depending upon the property of the cell is derived, it has different different types of applications that we can tune. So for instance, cell release heterogeneous of different size of cargoes. EVX cargo includes lipid, protein, and nucleic acid. So this can be used as therapeutics. So many more things. So this extracellular vesicle biogenesis is very unique. If we see here, there are different types of extracellular vesicles. Vesicles might be from the fragment of the cell surface, or vesicles might be from the endosomal origin here. So endocytocytes as well as exocytocytes to form different types of, basically when we lysate the cells, so it gives a different types of lipid assemblies. Those are all extracellular vesicles. So this nomenclature for extracellular vesicles, depending upon this origin, is different. So we call ectosome when it is fragmented from the cell surface. We also say from apoptotic bodies, so when it is fragmented by the apoptotic bodies, apoptotic cells. Similarly, the exosome, which is the unique in this extracellular vesicles, they are endosomal origins. So they basically these vesicles are generated within the endosomes, within the cell, and they are exocytocyte to the extracellular space. So these all are extracellular vesicles, so they have unique properties. So in our research, we started taking advantage of this extracellular vesicles, because one cell is releasing numerous numbers of extracellular vesicles. And these extracellular vesicles have numbers of proteins, as we have seen in this slide here, numbers of proteins. We really don't need all these proteins. The some proteins in the some synthetic nanoparticle will be tuned to target this nanoparticle to the intended sites. So that means the proteins from one extracellular vesicles can further engineer with the numbers of synthetic nanoparticles. That's the overall goal here. So we started our experiment with taking advantage of natural killer cell. So natural killer cell, by reading the surface receptor in this disease cancer cell, it basically finds automatically, naturally. And once it finds and senses the cells is infected, then it started to poke the hole on the surface and release the cytotoxic granules. And focusing on this mechanism of NK cell, what we did is we grabbed this, extracellular vesicle and this re-engineer with the liposome using different types of lipid here. So, and then a cytotoxic agent, we added here doxorubicin. So Because of the surface properties acquired from the extracellular vesicles, that's the mother of, from its mother, like an NK cell, it naturally seeks the, cancer cell. Okay? And now we have designed different types of fusogenic lipid, synthetic lipid in liposome that enhances the membrane fusion. And the, once it membrane fused, the internal loaded cargo will release into the cytosol of this cancer cell. So this is very unique, and then we are so happy. And Dr. Arun Kumar Tuyen and the Sagar is working on this direction. So how we do this experiment? So we isolate the vesicles, okay, by using different, centrifugation technique. So these vesicles are non-uniform. And then we started to use the liposome. So this liposome, as I mentioned, that our goal is to devise this nanotechnology, nanomedicine as a drug delivery as well as the imaging agent, so that it has multifunctionality. Okay. So we use the gadolinium label. We have the gadolinium phospholipid. So this phospholipid can be used to form the liposomes, and also use the doxorubicin extruded to the membrane extrusion and form the uniform biomimetic liposomes. So you can see here this NK-zome are heterogeneous in size, but by using the engineering technology, we can we can basically homogenize the size in this, the size range is what we want. So this one has aqueous core liposome. Similarly, this core can be tuned using the polymeric nanoparticle to form hydrophobic. Now this liposome system can use for water soluble drug, can be stored in aqueous core. Similarly, this hydrophobic polymer can be used to load the hydrophobic drugs. Okay, so once we synthesize this, engineer this nanomedicine, we started to learn whether we do have the properties of NK cell in these particles or not. So we use the signature proteins of the NK, whole cell lysate, as well as NK cell membrane and those vesicles. So we do found that all these signature proteins does present in the our engineered nanotechnology system. So because of this, presence, we do see the, specific targeting to the cancer. There's a difference in normal cell and the internalization of these particles and the cancer cell using FACS studies. So this gives us idea that, we are in achieving the target specificity of this, nanomedicine towards the cancer. Then after we started to look on the further details on this, in the cell using microscopic. So we can see that different signature proteins where in the cancer cell, we don't have CD50, we didn't find CD56, but in NK-zome, we do see CD56, which is NK-zome is the engineered nanoparticle from the, basically from the NK cell derived extracellular vesicles. And these particles are getting into the tumor. You can see the G stack. In the G stack, what we are seeing at a different plane of the cells, okay? And the blue is the nuclear staining, and this, green and the red is the rhodamine for the particles, and the green for the CD56 with the signature protein of the NK cell. So we do see that these particles, when internalized into the cell and into the nucleus, they are intact in the size. This is excellent. So this is basically, regular cell culture system where the cells are grown on the surface and nanoparticles is injected from the top, and we rely on the density gradient. So the nanoparticle goes to the monolayer of the cells and they find the receptor into cytosines. So that's basically density gradient. So we started to further think about the target specificity of this nanomedicine, and we learned, we basically we did the one, design one experiment, flow-based experiment, where the cells were grown here, but the nanomedicine, the nanoparticles was flow, towards this flow machine. So in such a way that when it is flowing, we can prevent the density gradient while we are maximizing the contact of the surface of the cell and the flowing nanoparticles. And we're so excited about the results that we have seen. So we can see that these nanoparticles were merged when it is in the cancer cell. Okay, so but different cell types has a different, different extent of the internalization. So when the bare liposome, the synthetic liposome without any extracellular vesicles, we do see the internalization, but this is very low as compared to these. These are very significant difference. And we saw that these particles has a very great, pharmacokinetics. So circulation half-life is increased because now our nanoparticle surface looks like immune cells, so our immune system can cooperate with this. So the regular nanomedicine has about 2 hours. So sorry for that. So 2 hours, but this one we found the 9 hours of circulation half-life, and we do see the high accumulation of tumor. Not only in this, ICP, we did the ICP analysis for this biodistribution. We do see in the section of the tumor that the particles are there. And we went to we studied the therapeutic potential of this nanomedicine, and we found that this medicine is, capable of reducing the tumor size. This is the 25 days, studies. So once the controlled tumor reaches at the certain size, we have to euthanize the animal following the IUK guideline. So this study is limited up to, basically up to 25 days. So, but the exciting news is we do see that release, decrease in the tumor size. Similarly, as I mentioned earlier, that we can tune this, surface from hydrophilic core to hydrophobic core. As you can see, this vesicles coated, polymeric nanoparticle. We do see the bilayers of the here, which is about 6 nanometer. This bilayer thickness in the cells are nearly 6 nanometer. What we found, nearly this coatings is a bilayer of the lipid. And we did that, studied the stability as well as the protein signature of this materials, and similar following in liposomal system. We saw that these particles were highly stable when it is coated, but when it is not coated, the PLG nano, without any coating, we do see the rapid aggregation. So this is the kinetics of the absorption at a 580 nanometer, where you will see the serum will aggregate, and this, with the particles, and you will see the, increase in absorbance. So this is a very unique experiment. And in the meantime, we also want to learn, because we have, we have encapsulated, gadolinium in these particles, because gadolinium has its own application as well as its own limitation. Gadolinium, when it is in the form of gadolinium ion, is highly toxic. But when the gadolinium is chelated in the macrocyclic chelator, as I have shown in earlier slide, then it is compatible. Okay, so we want to know whether this gadolinium is releasing very fast or not. So we do see some kind of, acid sensitivity in this, release pattern, but the release is still less than 10%, okay, which is excellent. And this particles does get into nucleus. We do found that these particles are getting into nucleus. That means they are targeting the cancer cell. Okay, so now we are ready to inject this particle to animal. Before that, we want to know what is the magnetic property of this particle, because we have encapsulated this macrocyclic chelated phospholipid, gadolinium phospholipid, into this nanoparticle system bilayer. So we saw that, we were able to enhance the magnetic relaxivity up to 5.3. So clinical magnetism is also the gadolinium contrast agent, which has 3. So our, engineer nanomedicine has a higher. So that means that to achieve the clinical contrast, we can reduce the injected dose of this nanoparticle. So which will help us to decrease or manage the, associated toxicity that gadolinium has. And we can, after injection, we do see the tumor lights up in this, a mouse. And also we do see the vascular, lightening up. So that can also be used as a vascular angiography, because the NK cells are we are using NK cells from the blood pool. So they can be tuned to the, vascular angiography. Similarly, since we added here gadolinium, we also added one near infrared dye in this, nanoparticle, and further understand that, to envision the tumor specificity of this particle. And we found that, this particle is, getting into the tumor with time. You can see here, as compared to the non-targeted particle, the control particle, which is distributed throughout the body of the animal. Here you can see, but this, engineered nanoparticle has, so to some extent, the precision in the tumor here. Of course, if they are the liver, spleen, those are the, phagocytic organs, we do see that. But once we dissect this tumor and we saw the differences in the tumor localization of this particle is highly significant. So up to this level, I have shown you how we can tune the natural killer cell properties to target or to design the nanomedicine that can target to the cancer. And the second part of this project, I would like to take the advantage of the work from the Sagar. So Sagar, right now, he's a postdoc at University of Kansas. So Sagar started to challenge himself and to further understand the deep into the extracellular vesicles using the macrophages. So what he did here is, he isolated the extracellular vesicle. Are you trying to minimize the time by using the unique, please follow this paper for this uniqueness of what are the techniques, the isolation techniques we have applied. So he's trying to minimize the, associated time for, extraction of isolation of extracellular vesicles. And he found that there are very nice, excellent, work done by Sagar. And once he has these vesicles out, he further infuse this vesicle with the synthetic liposomes and to form the hybridized liposomes, and which has a very precision in the tumor targeting, okay, tumor targeting, and also has a precision to the endosomal escape, because we want to protect this nanoparticle from the endosome, because endosome is highly acidic. It has potential ability to degrade all the material content in the nanomedicine. So that was the overall goal, because macrophage vesicles can have that opportunity to evade that in endosomes. Then we started to work on the targeting specificity of this engineered hybridized vesicles, which we call the hybridized gadolinium vesicles, which is hybridized with the macrophage derived extracellular vesicles. Since this is macrophage, we use a J774 mouse cell line. We do see the differences. That means that this particles also goes to the macrophage. The macrophage has eating property, right? And the cancer cell, we do see significant differences of these hybrid vesicles and will compare to liposomes. Similarly, another cancer cell also we do find differences. But when we go to the normal cell, normal fibroblast, we don't see significant difference. That gives us idea about the cancer specificity of this macrophage. Okay? And now, then after we started to know, okay, is this because of the macrophage or what really happening here? So we did one quick experiment. We treated these hybrid vesicles with the proteinase K. That proteinase K will basically chop down all the surface protein. We purify the particle and re-inject and retest them. So that means the in protein K positive, okay, that means protein that is proteins were chopped by proteinase K. Now particle becomes non-targeted. We do see some, now there is some. And proteinase K minus, the regular particles with intact protein on the surface, there is a significant difference. So that means that this protein, which are signature of extracellular vesicles, signature of macrophage, are working in such a way that these particles are specifically going towards the cancer. So which is outstanding achievement that we have done. And in this paper, we also discuss about how we can manipulate the isolation technique in the extracellular vesicles so that we can minimize our time. Then after, we have this gadolinium with a contrast agent we insert. Because of this phospholipid hydrophobic tail, we were very successful to insert this. In this case, we were very, very successful to enhance the relaxivity up to 9. So this is a tremendous but highly significant enhancement as compared to the clinical contrast agent. So that means we can reduce the injected dose to find the clinical level of contrast. So that will help us to minimize our toxicity associated with gadolinium. Remember, I would like to further enhance that. This gadolinium, when they are chelated within the chelation, they are biocompatible to some extent. But when this gadolinium is released as an ion, this is highly toxic. Gadolinium has lots of nephrogenic systemic fibrosis, NSF toxicity, organ fibrosis. It has a huge toxicity. That is also the concern that gadolinium need to be thinking about redesigning and revisit the formulations of currently used gadolinium. So I hope that this technology, that what we are right now presenting, what we are doing in our laboratory, might help us to understand the further move forward to revisit the current formulations in the gadolinium contrast agent media. So once we inject this into the next, we inject it, we saw this in very precisely in the blood vasculature as well as the targeting to the tumor. This gives us so excitement. Okay, so while this MRI experiment is very, as a new investigators, organizing and finding radiologists and taking their time, clinicians is really very difficult. So because they have clinical duties while they are helping a lot and they are encouraging bench scientists, but in the meantime, the finding the clinical time and the research time is quite difficult. So we started to think about if we can do some kinetics of these particles, that would be really great. But it's very difficult with the MRI because we need to take the sample objects, specimens out from the bed, and there are lots of things going on. So what we did is we started to think about differently and inserted the near infrared fluorescent dye in this nanoparticle, and we started to see that because we do have this instrument in our laboratory and more easy to work. So we do see these particles are getting into time, getting into the tumor. So you can see with time, you can see the tumor is more particles. These particles are green, labeled with green fluorescent dye. We do see the lots of particles are getting into the tumor. This gives us so excitement. So, and we are continuously working in this direction. So I hope with this all the the summary of the research that what we are doing in our laboratory, at right now at the UT Tyler, to give you some idea about the how we can manipulate, how we can re-engineer the properties of extracellular vesicles, how we can re-engineer the properties of synthetic vesicles. So the extracellular vesicles has some stability problem for long term in aqueous environment. Um, Synthetic nanoparticles can be redesigned to become highly compatible with the aqueous environment, but does not have targeting ability. The extracellular vesicles, when we start to load drug in our other experiment, we have stability issue. But we hybridize these two together. What is happening here? We are basically one is helping to other. So the property limitation of extracellular vesicles can be overcome by liposynthetic nanoparticles, and a limitation in synthetic nanoparticle can be overcome by the extracellular vesicles. So basically we are overcoming the respective shortcomings. So with this, I think I have showed you the robust biomimetic drug delivery carrier platform developed inspiration from the lipoproteins present in the cells. I have also explained about the integrating of the targeting properties from the extracellular vesicles and imaging contrast agent and so on. And also we discussed about the products of cells, what we call the extracellular vesicles, and we can envision these extracellular vesicles as a future nanomedicine. I hope that I was able to convince you the engineering techniques here, what we are using, simple liposomal fabrication technique. And there are still the works are ongoing, and I'm sure that there are lots of questions related to the immunogenicity of these particles, which we are continuously working on towards prediction. But this presentation is specifically focused on the what are the opportunities of re-engineering the biologics with synthetic nanoparticles. With this, I would like to thank all my collaborators and lab personnel. So most of the work today I present was done by the 2N, who is right now postdoc at MD Anderson Cancer Center. Ramesh Marasini, Dr. Ramesh, who is the postdoc at Johns Hopkins today, this time. And Sagar is the postdoc right now after completing. From this year, he's starting his postdoc at University of Kansas Medical Centers. And the undergraduate students, Caesar and Colin, as well as our research, visiting scholar, Jessica, they have lots of different level of involvement in this research. I would like to thank all our collaborators. Thanks to Professor Nancy Monterrey and Jim Revere, who have established the Nanotechnology Innovation Center at Kansas State, for which research I'm continuing here at the UT Tyler. Now I would like to thank the National Institute of Biomedical Imaging and by an entire NIS for the support for this research. So thank you very much. I'll be very happy to have any questions, suggestions. Thank you very much.